I mentioned why in our data we had a male preponderance in the behavioral readouts in our germ-free animals. And so that got us thinking about disorders where there's a higher male incidence, such as schizophrenia, but also autism spectrum disorders where there's 4-to-1 more prevalence. Also, autism has a very strong incidence of gastrointestinal symptoms. And talking to parents, that some of these GI symptoms are if we had some way of managing them, it would also be a very good thing in autism. But it's important to remember autism is a genetic disorder. We're not taking away from that in any means, but maybe there are ways to look at some of the symptoms of autism through targeting the microbiome. So we were interested in social behavior, and we wanted to see, well, what would happen if we took our germ-free animals and looked at how they responded to other animals. So this is using the 3-chamber test of Jackie Crawley, and mice, like humans, are quite social. So if you give them the opportunity to spend time with another mouse or an empty chamber, they will prefer to spend time with the other mouse, but not if they're germ-free. Mice, like some humans, are a little bit fickle, so if you give them the chance of spending time with someone they know or a new playmate, they'll prefer to spend time with the new playmate, but again, not if they're germ-free. We also found that there was increased repetitive behavior in these animals. So it tells us that for normal social responding, we need to have an appropriate repertoire of bacteria in our gut. And this is also in line with a growing body of literature emerging in other autism models that the microbiome is at play. This is work in the Valparate model from the Utrecht group and Danone research, and then from Sarkis Mazmanian and Paul Patterson, the late Paul Patterson. They showed that in their maternal immune activation model that there was a disrupted microbiome, and that they could reverse this with a single bacteria, Bacteroides fragilis, and were able to elucidate some of the metabolites induced by that, which led quite, I think, still a little bit provocatively and prematurely, but the concept was published in Cell that we could develop probiotics for autism and other neurodevelopmental disorders, and they put forward this Jack Gilbert and Rob Knight put forward a way forward for doing that. But it does tell us it's an exciting area to look at some of the symptoms, both core and comorbid symptoms of autism. It also got me thinking of why on earth would you need bacteria in your gut for normal social behavior? Like, what is the evolutionary basis for this? So I collaborated with Seth Bordenstein, who's an evolutionary microbiologist at Vanderbilt, and because I was beginning to think, well, what's in it for the bacteria? And when you think of evolutionary theories, and the good thing with working with Seth was he was able to always set me back and say, John, you have to remember that the bacteria were there first, and that all of brain development has occurred in the context of the bacteria and the microbial milieu. And so we know that social development, going back to the Dunbar theories, is good for brain development and cortical elaboration, but perhaps the microbes have helped shape that as well in evolution. It's speculative, but it's quite, sometimes interesting to always put things in an evolutionary perspective. So this is, kind of one of my take-home slides. For those of you falling asleep, it's good to wake up right now, because it really is, we put this out in a Trends journal last year just to reinforce where we're going with this. In trying to understand the complexity of the brain across the lifespan, we're getting to grips with what's going on, and we're understanding the plasticity in the brain as we mature and as we age, and the critical periods in early life, adolescence, and old age. And you kind of can juxtapose these with the onset of a variety of mental disorders and a variety of different processes going on in the brain in that period. But I would propose that we also put the microbiome and changes in the microbiome that are occurring at these key periods, driven by diet and environment, etc., and look at these interactions at these critical periods is going to be very important for how we move forward and how we understand what's going on in the brain at these critical periods. And that it's important to remember that barrier function and maintaining both gut barrier as well as blood-brain barrier function will be important for maintaining maintaining the correct immune status, and we know that certain bacteria, we have probiotics, can actually help tighten barrier function, and so that's a big exciting area overall. Back to Metchnikoff, as I said, he's the father of probiotic research, and a probiotic, as everyone in this audience will know, is a live microorganism. When administered in adequate amounts, will confer a health benefit. So most of the things we call probiotics are not really probiotics because they haven't been proven to have a known health benefit. This is a yogurt from Korea. They have Metchnikoff on it, so it has that Nobel sign of approval. And so we were interested, given that stress affects your microbiome, and the germ-free study showed the microbiome affects stress, can we give a bacteria to modulate how we deal with stress? And we did this first in animals. And this is data from a few years ago where we gave a Lactobacillus rhamnosus. We tested these animals on the elevated plus maze, a well-known test of anxiety. If animals are more anxious, or less anxious, or given like Valium, they'll come out and spend time here. Or if they're in the swim test and they're given drugs like fluoxetine, Prozac, they'll spend more time in active behavior. And so basically our animals that have been given this Lactobacillus rhamnosus for a number of weeks behaved as if they were already on Valium or on Prozac. And so that's behavior. What about physiology? When we stressed these animals, we got a dampening down of their stress response. They were more chilled out. When we looked at their brains, we looked at the composition of GABA receptors across a variety of different brain areas, and you see wide-scale changes in GABA A and GABA B receptors. And so it tells us that by taking healthy mice, taking a specific lactobacillus can really affect their behavior, their physiology, and their neurochemistry. And the question is how? By what means? And is it specific to this bacteria? Here's data, more recent data with bifidobacteria. This is a biflongum and a bifbreve. We look at this defensive marble burying. If the animal is less anxious, it'll bury less of these marbles. And here you can see this is the animals here have an anxiolytic effect. We look at cognition. We looked at these animals that are allowed to find an escape route in this Barnes maze. And if they've been fed this bifidobacteria, they were able to make less errors, so it improved their working memory. And so all of this is like, well, it's fine, but what are the mechanisms at play? But the question is mechanism. And I showed you this picture with the vagus nerve. And so together with John Bienenstock's lab in Canada, we performed vagotomy, and we cut this communication highway. And to make a long story short, all of the effects on behavior and neurochemistry that we had seen were cut, were ablated when we cut the vagus. So it tells us, as I like to say, what happens in Vegas will affect your emotions. And that's an important thing to remember.